Hi everyone, I'm gonna discuss the process of mitosis and meiosis. Mitosis is a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus, typical or ordinary tissue growth. Mitosis is a type of cell division in which one cell, the mother, divides to produce two new cells, the daughters, that are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. The great majority of the cell divisions that happens in your body involve mitosis. During development and growth, mitosis populates an organism's body with cells, and throughout an organism's life, it replaces old, worn-out cells with new ones. Phases of mitosis. Mitosis consists of four basic phases, and these are prophase, metaphase, anaphase, and telophase. Prophase. The first and longest phase of mitosis is prophase. During prophase, chromatin condenses into chromosomes and the nuclear envelope or membrane break down. In animal cells, the centrioles near the nucleus begin to separate and move to opposite poles of the cell. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. Metaphase is a stage of mitosis in which eukaryotic cell cycle chromosomes are at their second most condensed and coiled stage. At this stage, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells, long protein filaments called kinetochore microtubules extended from poles on either end of the cell and attached to the kinetochores. Anaphase. Anaphase is the process that separates that duplicated genetic material carried to the nucleus of a parent cell into identical daughter cells. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. Telophase. Telophase is the final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of the parent cell into identical daughter cell. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cells. Meiosis. So what is the meaning of meiosis? Meiosis is a type of cell division of germ cells in sexually reproducing organisms used to produce the gametes such as sperm or egg cells. It involves two rounds of division that ultimately result in four cells with only one copy of each paternal and maternal chromosomes. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces four gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. Meiosis begins with the parent cells that is diploid, meaning it has two copies of each chromosome. There are six stages within each of the division, namely prophase, metaphase, anaphase, telophase, and cytokinesis. Meiosis 1. In meiosis 1, homologous chromosomes are separated into two cells such that there is one chromosome. per chromosome pair in each daughter cell. In meiosis 1, chromosome in a diploid cell resegregate, producing 4 haploid daughter cells. Prophase 1. Prior to prophase 1, chromosomes replicate to form sister chromatids. During prophase 1, homologous chromosomes pair and form synapses, a step unique to meiosis. The paired chromosomes are called bivalent, and the formation of chiasmata caused by genetic recombination becomes apparent. Chromosomal condensation allows this to be viewed in the microscope. Prometaphase 1. The second stage of the phase meiotic division, during which the nuclear envelope breaks down, allowing microtubule access to chromosome. During prometaphase 1, microtubule attach at the chromosomes kinetochores and the nuclear envelope breaks down. Metaphase 1. In metaphase 1, the homologous pair of chromosomes align on either side of the equatorial plate. Anaphase 1. In anaphase 1, the homologous are pulled apart and move apart to opposite ends of the cell. Each of the homologous chromosomes get pulled towards opposite poles of the cell as the spindle fibers retract to divide the DNA between the two cells which will be formed. Telophase 1 and cytokinesis 1. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. During telophase 1, the nuclear envelope reforms and spindle fibers disappears. In cytokinesis 1, the cytoplasm and cell divides, resulting in two cells that are technically haploid. There is one chromosome and two chromatids for each chromosomes. For each. Meiosis 2. During meiosis 2, the sister chromatids within the two daughter cells separate, forming four new haploid gametes. The mechanics of meiosis 2 is similar to mitosis, except that each dividing cells has only one set of homologous chromosomes. Prophase 2. During prophase 2, chromosomes condense and the nuclear envelope breaks down if needed. Prometaphase 2. During prometaphase 2, the nuclear envelopes are completely broken down, and each sister chromatids forms an individual kinetochore that attach to microtubules from opposite poles. Metaphase 2. In metaphase 2, the chromosomes line up individually along the metaphase plate. Anaphase 2. In anaphase 2, the sister chromatids separate and are pulled towards opposite poles of the cell. Telophase 2 and cytokinesis 2. In telophase, the nuclear membrane forms around each set of chromosomes and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final products of meiosis, four haploid cells in which each chromosome has just one chromatid.